Assalamualaikum warahmatullahi wabarakatuh. I am Wahyuni Ramadhani Fitri, NPM H1A021002, a medical student, Faculty of Medicine and Health Sciences, University of Bengkulu, with Honorable Selector Dr. Wisma Yunita, MPD. I will present a medical journal article, case report, prenatal diagnosis of a fetus with harlequin ichthyosis identifies novel compound heterozygous variants, a case report, by Jiao Liu, Xingyu Zhang, Weilan Wang, Xiaofang Lan, Minyu Dong, Kai Yan, Yongliang Lei, Penglong Chen, Mufeng Yang, Kundasan, and Chunlei Jin. Harlequin ichthyosis is the most severe form of the keratinizing disorders, and it's characterized by whole-body head-stratum corneum. ABCA12 has been identified as the major disease-causing gene of harlequin ichthyosis. The aim of this study is to identify new heterozygous variants through prenatal molecular diagnosis of genetic counseling cases. Inherited ichthyosis are a group of genetic defects characterized by generalized dry skin, scaling, and hyperkeratosis. Harlequin ichthyosis is the most severe form of ichthyosis. It is a rare genodermatological disease. Harlequin ichthyosis is an autosomal recessive disease with an incidence of 1 in 300,000 births. Pathogenic variants of ABCA12 have been demonstrated as the major causes of harlequin ichthyosis. The ABCA12 protein function as a lipid transporter, transferring lipids from the cytosol into lamellar granules in healthy skin. The lamellar granules fuse with the cell membrane and release their content into the intracellular lamellae. In the skin of harlequin ichthyosis patients, dysfunctions of ABCA12 results in disordered lipid transfers. As a consequence, abnormal lipid-containing fake walls form in the corneocyte cytoplasm. The skin turns into a defective formation of the lipid layer, and the stratum corneum grows remarkably thickened. The clinical feature of harlequin ichthyosis is characterized by markedly thickened stratum corneum covering the whole body, which cracks and forms erythematous fissures soon after birth. The skin abnormalities also affect the shape of eyelids, ears, nose, and lips, and babies with this congenital skin disorder are usually born prematurely. A case of harlequin ichthyosis was prenatally diagnosed by ultrasonography and genetic tests. The fetus had been found with interfacial deformity and profound thickening of the palm and plantar soft tissues. Chromosomal microarray analysis (CMA) and whole exome sequencing (WES) were then performed on the amniotic fluid to identify germline pathogenic variants for the fetus. Candidate variants were verified by Sanger sequencing. Genetic counseling was provided to a 29-year-old Chinese woman during her gestational 23 weeks since she had an abnormal fetal ultrasonography results. The woman delivered a healthy female baby at term during her first pregnancy. Figure 1. Clinical presentation of the proband. A. Degree of the family. Transmission of the compound heterozygous variants of the harlequin ichthyosis baby were labeled in the 3 generations family. B. The baby was induced by vaginal delivery at 23 weeks of gestation. The clinical picture of harlequin ichthyosis infants is that the fetus is wrapped in a thick, plate-like collodion membrane over the entire surface of the body at birth. Moderate nutrition, no obvious eyelid atrophy and undeveloped eyeball. The distance between the eyes. Nasal collapse. Nasal bones show poor development. The mouth and lips are curved and swollen, cleft, with prominent alveoli and short mandible. Bilateral auricle development is poor and the auditory canal is present. The skin of the whole body is covered with light yellow horns. Underdeveloped hands and feet. Fingers and toes bend unlike claws. Poor development of external genitalia. Gender discrimination is not clear. Male considerations. And there is anus. Pathological examination of the skin reveals severe epidermal hyperkeratosis. Ultrasonography at the 12th week of this pregnancy indicated that the fetus had multiple malformations such as a short face, abnormal nasal bone, ear and mandible, and a cleft palate. At the 16th week of pregnancy, the fetus showed prevalent thickening of the palm and plantar soft tissues according to ultrasonography. The clinical diagnosis was not determined based on ultrasonography. Subsequent genetic tests were performed to make a definite diagnosis for the fetus. Combined with the results of whole exome sequencing, WES, and ultrasonography, the fetus was finally diagnosed as harlequin ichthyosis. Amniotic fluid is collected for genetic analysis, Sanger sequencing verification of the harlequin ichthyosis compound, heterozygous variants identified by WES in the family. The research suggested that the sister, the mother, as well as the maternal grandmother carried the heterozygous C2157-delta, and the father and the paternal grandmother carried the heterozygous C6858-delta. B. Summarizations of published of harlequin ichthyosis variants. The harlequin ichthyosis variant that alters the ABCA12 protein sequence. The ABCA12 gene has been characterized as a major disease-causing gene for harlequin ichthyosis. ABCA12 is a member of the adenosine triphosphate binding acid (ABC) superfamily of active transporters. Variants in the ABC gene cause various diseases such as cystic fibrosis, Danger disease, pseudoxanthoma elasticum, and Dubin-Jansen syndrome. The dysfunctions of the ABCA12 protein can directly affect the formation of the lipid layer between cells, which is important for the function of the epidermal barrier. The prenatal ultrasound diagnosis of harlequin ichthyosis was confirmed by molecular diagnosis and skin biopsy. Through WES, we identified two inherited heterozygous friendship variants that would be helpful for the diagnosis of harlequin ichthyosis in the future. Compound heterozygous friendship variants of ABCA12 were identified for the fetus, suggesting the former variants were maternally inherited and the latter paternally inherited. The fetus were terminated. A prenatal molecular diagnosis is an important approach for the prevention of harlequin ichthyosis. In the study, we provided that a successful case of genetic counseling for a family with an harlequin ichthyosis baby.